Thank you for standing by. Today's conference will begin momentarily. Again, thank you so much for standing by. Today's conference will begin in just a moment. Welcome.
Hi, good afternoon. I'm Monica Witt from NASA's Office of Communications.
Oh,
Thank you for joining us.
Monica.
Oh.
Just one moment, Ms. Monica. I do apologize.
Oh.
Welcome, and
No
thank you
worries.
for standing by. At this time, all participants are in a listen-only mode. During the Q&A session, if you would like to ask a question, you can press star 1 on your phone. Today's conference is being recorded. If you happen to have any objections, you may disconnect at this time. And now I'd like to turn it over to Ms. Monica Witt. You may begin.
Hi, good afternoon. I'm Monica Witt from NASA's Office of Communications. Thank you for joining us today to talk with experts about science investigations launching on Northrop Grumman's 15th commercial resupply. Launch is targeted for Saturday, February 20th from the Mid-Atlantic Regional Spaceport's Pad 0A at NASA's Wallops Flight Facility on Wallops Island in Virginia. A 5-minute launch window will open at approximately 12:36 p.m. Eastern. The backup launch will be on Sunday, February 21st at 12:14 p.m. Eastern. Northrop Grumman is scheduled to deliver about 8,000 pounds of cargo to the orbiting laboratory, including critical supplies in support of some of the 200 to 300 experiments that are ongoing at any given time aboard the space station. Here to talk with us today about some of the science launching aboard this mission are the experts. For the Spaceborne Computer 2, Dr. Mark Fernandez. For the protein-based artificial retina manufacturing investigation, Nicole Wagner, President and CEO of Lambda Vision, and Jordan Greco, Chief Scientific Officer of Lambda Vision. For the Micro 16 experiment, Dr. Shiva Vanapalli, Professor of Chemical Engineering at Texas Tech. To talk about the real-time protein and crystal growth 2 study, Victoria Drago, University of Toledo, PhD candidate. To discuss the AHOS, a radiation detection system developed for the Orion spacecraft and certified for use on NASA's Artemis II mission, Carrie Lee will be joining us. Our speakers today will provide a brief overview of the research, and we will open the lines for a few questions after each speaker. If we have time at the end, we will open up lines for additional questions. Your phones are on mute now. To get into the question queue, you can press *1 on your phone at any time. The operator will call on you and open your mic so that you can ask your question. We ask that you please stick to one question, and if we have time, we'll allow reporters to ask a second question. About 1 hour after we conclude, you can listen to a replay of this teleconference by dialing toll-free 800-925-0147. One more time, to listen to a replay about an hour after the teleconference, you can dial toll-free 800-925-0147. Thank you. To start, we will hear from Heidi Paris, Associate Program Scientist for the International Space Station Program at NASA's Johnson Space Center in Houston, for an overview of some of the science going up on this mission. Over to you, Heidi.
All right, thank you, Monica. Good next week. Before we get started talking about the NG-15 research, though, I wanted to start by talking a little bit about trees. So I grew up in Northern California under the canopy of these, giant, beautiful, towering redwood trees. So trees hold a really special place in my heart, and I think folks who know me know that I tend to think about the world in general in terms of trees. And I think trees are also a really good metaphor for the research that's being done on the International Space Station. So every broad area, every collection of research that we do on the International Space Station is kind of like a branch on our tree, right? And every individual experiment is like a leaf. And we have dozens or sometimes even hundreds of leaves on each branch that are representing the things that we're continuing to study and to learn in that area of research. So the more research that gets done on the space station, the bigger our tree grows, the more we learn, and the more that we have these kind of offshoot branches developing for new areas of research that are based on the insights that we gain from previous research that we've done. So we just actually saw a new offshoot branch kind of sprout up over the last couple of weeks on the ISS. We have plant researchers who are currently attempting to grow vegetables in space, right, which it turns out is very difficult to do in a microgravity environment. So when some of these plants weren't really growing all that well, the science team talked about it. They decided to have the crew try a pretty risky transplant procedure, which in the current state of growth of those plants, it rarely works even on the ground, let alone with the challenges in microgravity. But surprisingly, it worked great, and the vegetables are thriving now. So the question that we ask is, why did that work so well? And we have no idea yet, but if we can find out why, it might have huge impacts to how well we're able to grow food during future long-duration space missions, and that is going to be extremely important for exploring outside of low Earth orbit when we go, onto moon, to the moon and then onto Mars as well. So these kind of exciting discoveries really are happening a lot on the space station. And that's one of the reasons that I believe if we are thinking of ISS research like a tree, then really ISS research is in full bloom right now. There are hundreds of different experiments going on simultaneously on the International Space Station, and it's putting into motion the work of some of the most brilliant scientific minds in the world. And each piece of the puzzle, each leaf on our tree, it's attempting to make our daily lives better on Earth, and also to continue to expand our ability for future space exploration. So we have lots of really great science coming up on NG-15 next week to continue to grow our research tree and expand our knowledge. You'll hear about some of those from the researchers themselves today, which is always so much fun. These men and women are super passionate about what they do and very dedicated to their research work. Several of the investigations that we'll talk about today, including Spaceborne Computers 2, the protein-based artificial retina manufacturing investigation, and also the real-time protein crystal growth 2 investigation. These experiments all represent kind of those offshoot branches on our research tree. So they are follow-up experiments that are taking what they learned from a previous investigation and now kind of taking it up to the next level and seeing what more they can learn and how we can use that technology in an even better way, more advanced way. We'll also hear from the scientists for the Micro 16 experiment and the AHOS experiment. These two experiments are studying two of the most significant challenges that we face in long-duration spaceflight. And those are muscle weakening. Talk about just one reference that I'll leave you with. If you're interested in more information about ISS science, a great place to find that is on our website at www.nasa.gov/ISS-science. And you can also follow us on Twitter at ISS_research. So Monica, back to you.
Thank you so much, Heidi. For the first investigation, we'll hear from Dr. Mark Fernandez, the Spaceborne Computer 2. Dr. Fernandez is the principal investigator of Spaceborne Computer 2 and solutions architect for Converged Edge Systems at Hewlett-Packard Enterprise. Dr. Fernandez?
Well, thank you very much, Monica. I appreciate being here, and I appreciate everyone on the line. So I'm Mark Fernandez. I'm the PI of Spaceborne Computer 2. And as you can imagine, it's a follow-on to Spaceborne Computer 1. With Spaceborne Computer 1, we proved that we can take modern, commercial, off-the-shelf, state-of-the-art, enterprise-class computers to space on the ISS in what we call the edge of the edge, if you will. The need to do that when we get to Mars was beautifully illustrated in the movie The Martian. You saw that Matt Damon was sitting in the red dust of the Mars planet, and it took 24 minutes of communication delay for him to get information back to Earth. And then once they solved his problem, it took 24 minutes to get back. And he literally jumped up and was celebrated at almost an hour later when he got his answers. Well, when we get to the moon and we get to Mars, we're going to want to take our modern computers with us, A, to avoid those delays, and B, preserve the limited bandwidth back to Earth. So we need to be able to prove that we can do that. With Spaceborne Computer 1, NASA and Hewlett-Packard Enterprise worked together, and that's what we focused on. And it was a success. We were able to package up unmodified computers right off the factory line and have them survive the shake, rattle, and roll of a launch. They were successfully installed by the astronauts, and we were in successfully for 1.8 years and got the correct answers. Now, Spaceborne 2 is going up on the 20th on NG-15, as has been presented, and I want to focus on what I think is the most important aspect of our mission, and that is it is not for us or for NASA or the ISS National Lab. It is for the community, the research community, to explore taking advantage of these computing capabilities in space at the edge of the edge on the ISS. So to augment the hardware that we sent up with Spaceborne 1, we're including HPE Edge-Line Converged Edge Systems, which include GPUs that are designed and purposely engineered for edge computing and to do artificial intelligence and machine language processing. I constantly tell myself that the purpose of research and exploration is not data collection, but insight. And we want to get that insight out to the researchers as quickly as possible. So we want to propose to the community, do you have an experiment where the time it takes you to get your data back to Earth and then process it here, is that shorter than it will take the time to process the data on Spaceborne Computer 2 at the edge of the edge on the ISS, plus the download time of that smaller output, hopefully insightful output data that you've created. So that's what we want to do. You can find more information about Spaceborne Computer and how to apply to take advantage of this research opportunity at hpe.com/info/Spaceborne. I think the introduction was very good, and I'm hoping that with Spaceborne Computer 2, we can help grow the whole community and build a stronger tree. And with that, let me hand it back to you, Monica. Thank you.
Thank you, Dr. Fernandez. If reporters would like to ask a question about Spaceborne Computer 2, please press *1 on your phone. The operator will announce you and open and close your mic for you to ask your question. Again, please try to stick to one question, and if we have time, we'll allow a second question for each reporter. Thank you.
All right, and we do have the first question coming from Camden Hall from Nashville News. Your line is now open.
Hi there. Thanks for taking my question. My question, I'm not sure if this one would apply to you, but it's about the Antares vehicle. I know that you all use RP1 and LOX on the first stage. Could you tell us a little bit more about why the second stage does not have that? Again, I'm not sure if that's a question for you or somebody else later in the briefing.
That wouldn't be for Mark Fernandez at HPE with Spaceborne Computer 2. That's more with the vehicle.
Okay, thank you.
Okay, and as another reminder, if you'd like to ask a question, please press *1 and record your name clearly and your affiliation.
I have a quick question for you, Dr. Fernandez. I'm curious, how and what can we learn by using the Spaceborne Computer 2 in space, and how will it be applied back here on Earth?
Thank you for that question. So artificial intelligence and machine learning were in their infancy when we had Spaceborne Computer 1 going up in 2014 to 2017. AI and machine learning and edge computing are maturing now, and the edge is everywhere. We've got a potential partner that wants to run an experiment on Spaceborne Computer 2 on the ISS so that they can then tell his customers, hey, if I can run in space, I can run anywhere, whether that's on your offshore oil rig, whether that's in the depths of your coal mine, whether that's on a high-speed train, whether that's it's your drone surveilling crops or trees or cities. So as we advance and prove out the ability to compute at the edge and deliver those insights back to the scientists that need them in their offices and homes, then that improves the aspects for all of humanity. Thank you.
Okay, and we do have another question. It comes from Kenneth Chang from New York Times. Your line is now open.
Great, thank you. It sounds like the bottleneck is sending data to and from the station. So what is, how much data can go up and down? And I guess how does that limit what can be done on the ground? And I was wondering, is a computer at all different that is in space? Thank you.
So that's another good question. A colleague had a good analogy yesterday that he gave me. He said, our cell phones, our televisions, our cameras are all increasing their resolution at incredible rates. But we all sit there and wait, when am I going to get 5G? When am I going to get 6G, etc.? That the network between my devices, my cell phone, my Netflix connection, etc., is the bottleneck. And it's the same way coming down from the ISS. When we're interacting with our computer on the ISS, we have a latency of 700 to 900 milliseconds. And if you're as old as I am, that's like going back to the modem days. You type a character and it eventually shows up. So technically we have 2 megabits per second up and down, and that compares to about 50 megabits a second that I have from AT&T in my home. So to help with that, you want to do the computing at the edge and minimize the download size. We will use encoding and compression furthermore, to improve that time that you spend on the network before you get your results or your insight. Does that make sense?
Thank you, Dr. Fernandez.
Mm-hmm.
Next, we will hear from Dr. Nicole Wagner and Dr. Jordan Greco. Dr. Wagner is President and CEO of Lambda Vision, and Dr. Greco is Chief Scientific Officer of Lambda Vision, a startup company sending their second experiment to the space station. Dr. Wagner has played an integral role in the founding and development of Lambda Vision's protein-based artificial retina. Dr. Greco's research has focused on studying light-activated proteins and their application into devices and drug products, which has led to the development of Lambda Vision's artificial retina and the production techniques currently under investigation on the ISS. Dr. Wagner, would you like to start?
Great, thank you very much. So I will start by giving a brief overview of Lambda Vision and what we're doing, and then I'll turn it over to Jordan to talk a little bit about what we're going to be doing on NG15. So Lambda Vision is a startup company that is developing a protein-based artificial retina to restore vision to the millions of patients that are blinded by end-stage retinal degenerative diseases. We will target the orphan indication of retinitis pigmentosa first, and then we'll follow on to the much larger market of age-related macular degeneration. Now, while there's some treatments for these diseases, most of these treatments only slow the progression of the disease, and people are going blind. Lambda Vision's technology is a small, simple, flexible implant, which will be placed in the back of the eye through a simple surgical procedure, something very similar to a retinal detachment procedure. And the goal is to replace the function of the damaged rods and cones of the eye. So these are your light-sensing cells. And our artificial retina will provide high-resolution vision to these patients. Now, you may ask, how do we get started with working with ISS National Labs? So we started in 2016 when we won the CASIS Boeing Prize as part of the Mass Challenge Accelerator Program. And so the goal of this project that we were awarded funds for was to explore the effects of microgravity on the layer-by-layer deposition process used to create the artificial retina. Now that process right now that we're doing terrestrially is very much subject to the effects of gravity. So things like sedimentation, surface tension, and evaporation can all lead to irregularities in the layering process, which can lead to detriment in the artificial retina. So in the past several years, we've worked very closely with our implementation partner, Space Tango, to build a layer-by-layer system for use on the ISS, which we actually flew on SpaceX 16 in 2018. Now, the results from that initial feasibility study were very encouraging, and it supported us to apply for a phase 1 and phase 2 SBIR with NASA to further look at parameterization of that experiment, to improve the layering process, and also to explore the layer-by-layer process for applications even beyond the artificial retina. Finally, we were awarded a NASA NRA in June of 2020 to look at production processes, which include establishing good manufacturing capabilities for work on the International Space Station. Now, this is really important because what we're doing in terms of these processes and procedures will be critical for anything that needs to be manufactured that will have clinical application to people here on Earth. Jordan, do you want to talk a little bit about NG15?
Sure. Thanks, Nicole. On the upcoming NG15 flight, Lambda Vision and Space Tango will build on the insights gained during the first Pathfinder mission on SpaceX 16, which Nicole just described. In addition to years of terrestrial-based research on the layer-by-layer production of artificial retinas. So much has been learned during the last 2 years, both with respect to our research facilities in Lambda Vision and also in Space Tango, and through the collaborative process between the two entities. And the latest manufacturing apparatus that's going to be contained on the Cube Lab represents many important advancements in the engineering and hardware required for spaceflight for this mission. So on NG15, really the focus is hardware and software oriented, centered on characterizing fluidics, operational controls, in-process quality controls. They're all necessary for successful production of the artificial retinas in low Earth orbit. So this is the second of what we expect to be many flights And we view this and each flight as iterative, an important part of the research and development process. This particular layering trial will allow us to continue to gather critical information on the design of the system and to continue to probe the influence of microgravity on this layering process that we've described. In addition, and very importantly to note, this is also helping us build the necessary controls required to support good manufacturing processes and other regulatory requirements for producing the artificial retina. So we're excited to continue this partnership with Space Tango, NASA, and the ISS National Lab, and also just very thrilled to have a chance to establish a foundation for producing products in low Earth orbit with true clinical benefit for patients, and in our case, for patients that are blinded by these devastating retinal degenerative diseases. Thank you.
Thank you, Dr. Wagner and Dr. Greco. We will now go to reporters for questions. Please press *1 on your phone to be entered into the queue.
Okay, and our first question comes from Bill Howard from CBS News. Your line is now open.
Thank you very much. For someone who doesn't follow this sort of technology, can you say a few words about how it interfaces with the optic nerve? How do signals get to the brain? I mean, I don't understand the basic biology we're talking about here with an artificial retina. Thanks.
Sure, I'm happy to take that question. So what happens if you have retinitis pigmentosa or macular degeneration is that you lose the light sensing cells of the eye. So these are your rods and cones. So essentially what happens is your eyes become insensitive to light, so they can't take the light in the room, and convert it into a signal that can be sent to the optic nerve and then to the brain. So what our implant does, it's a very simple structure. We have a scaffold, we have a polymer, and then we have a protein, and we do that multiple layers. And so that structure we actually put into the same space where those cells would exist, your rods and cone cells would exist. And in response to light, now the protein is going to take the light and it's going to convert it into a signal that's going to be sent to the optic nerve in the brain. So you can really think of this artificial retina as an artificial photoreceptor cell layer, where we're replacing the function of the damaged rods and cones. So we're absorbing light and generating an ion gradient in response to that light. So our artificial retina will interface directly with the remaining neural circuitry in the eye.
Okay, and our next question comes from Mark Carey from Aviation Week and Space Technology. Your line is now open.
Thank you. I'm wondering if this is a data relay to the ground experiment, or do you have tissues and materials that have to come back? And if that's the case, when and how? And also, if I might ask a related question, is this something, a technology that's sort of on the horizon, or could this be like many years away from materializing for use on Earth?
Sure. So I can answer that as well. So the artificial retina that we're developing right now, the work that we're sending on NG15 is we're sending the protein materials, and we're actually going to manufacture the artificial retina on the ISS. So we're doing this layer by layer process on board the International Space Station, and then those films will then be returned back to Earth for analysis. Though, even though that's the case, we also have real-time processing as well. So Space Tango is getting feedback through imaging that is allowing us to view the thin films during the whole production process. So we do have some control there. In terms of the development timeline, we are targeting, hopefully being in the clinic within the next 3 to 4 years. And I think part of this process is obviously the manufacturing, which we're working very diligently towards right now, and it's a big part of this.
Great. Thank you, Dr. Wagner and Dr. Greco. Next up is Dr. Shiva Vanapalli, Principal Investigator for the Micro 16 investigation. Dr. Vanapalli is a professor of chemical engineering and the Brian Pierce Bagley Regents Chair at Texas Tech University. His research lab works in the area of microfluidics, cancer, and healthy aging. Dr. Vanapalli?
Hi, thank you. I appreciate the opportunity here. I guess initially I'll just give a brief overview of the experiment. So our project here is focused on looking at the strength changes in this nematode across across 8 generations. So as you all probably know that in long duration experiments or in long duration in this long duration explorations, a significant challenge is the astronaut health, and the strength of the astronauts declines as they spend a significant amount of time on the ISS. So as a result, there is a significant need to understand the actual biology which is responsible for this in terms of this loss in strength. And to understand this biology, our project is focused on taking these nematodes or worms and looking at how the strength of these worms is changing. And this project really is at the intersection of engineering and physics and biology. And I'm truly excited because it really brings all these disciplines. And I would like to elaborate on this. So in terms of the animal here, so this it's really small, and if you want to measure how strong it is, you need to really figure out a way to measure all the forces which this worm is exerting. So our lab and students have built this device which essentially has an array of soft pillars, and the worm, as it crawls through these array of these pillars, we can record pictures of how much is the deflection which is happening. So as a result, by recording all the deflections, we can measure the forces which this animal is actually exerting. So our hypothesis is that previous experiments with this animal have shown significant changes in the genes. However, and we don't know if those changes in genes actually translate to any changes in strength. So our device essentially allows us to measure the strength, and also we'll be able to obtain the data on the changes in the gene expression. So it will really inform us how these changes in gene expression lead to changes in strength. And the other important aspect of our experiment is that these animals, and they live about 3 weeks, and in roughly about a week, and they produce all this progeny. So now we are interested in seeing if those progeny also see similar changes in the gene expression and the strength as well. So in that sense, it's very exciting to understand how, in a given species, how basically 8 generations are witnessing any changes in the expressions of genes and the strength loss. Now, this is in terms of our experiment on the ISS. However, if this experiment shows that we do indeed observe a strength loss, then it provides an easy platform to screen drugs, because as you probably know, and we have significant diseases in which the impairment of the muscles happen. So as a result, if we do observe that our device is able to record these changes in strength, then it opens up tremendous opportunities in conducting experiments on different drugs and figuring out how to maintain and improve the health of astronauts. Again, I'll be happy to take any questions about this experiment or some details if you like.
Thank you so much, Dr. Banapalli. We will now take questions. Again, please press *1 to be entered into the queue. I have a quick question, Dr. Banapalli. What are some of the unique aspects of the Micro 16 spaceflight experiment?
Sure, yeah. So in terms of the, in terms of the innovation here is that, we'll be taking about 100,000 images of these worms which are deflating all the spillers. And this visual data would be a really tremendous resource in terms of course, understanding the actual biology of this animal. How, in terms of education and outreach, we're actually planning activities in which high school kids and so on could actually use these images and learn about the engineering and physics and the biology aspects of this project. So in short, in terms of the novelty, is the engineering aspect of this device, which can measure the strength, and also we had to figure out a way to measure the strength of the same animal age. As you might appreciate, older animals will have strength which is less as compared to animals which are of a younger age. So in that sense, we basically have to figure out a way to synchronize animals' age on the space station so that we obtain strength of the same aged population.
Great, thank you so much. Next, we will hear from Victoria from the Real-Time Protein Crystal Growth 2 experiment. Victoria is a fourth-year PhD. Victoria?
Hi, thank you. So I want to start by saying that this is my third and our lab's fourth flight to the ISS doing crystallization experiments. While our previous flights using our capillary dialysis apparatus were very successful in growing large crystals for neutron crystallography, the apparatus is not meant for large-scale production and does not allow for real-time monitoring. The Real-Time Protein Crystal Growth Initiative was implemented by astronaut and crystallographer Larry DeLucas, and he used 10-egg lysozyme on RT-PCG1. RT-PCG2, or Real-Time Protein Crystal Growth 2, is a unique opportunity that will allow us to grow, monitor, and optimize protein crystals in microgravity through real-time communication with ISS crew members. The real-time communication with the ISS crew members is what really sets us apart from previous microgravity crystallization experiments. On Earth, unit gravity induces convection currents and thermal shearing that may hinder crystal growth and makes crystal growth feeding irregular. This often results in defects or inclusions that affect data collection. The primary advantage of microgravity crystal growth is producing large crystals with highly ordered lattices. We have prepared two proteins for these experiments. The first protein, which we're using as a model protein, is sperm whale myoglobin. The sample we were flying was recovered from a 1963 ammonium sulfate paste prepared by Alan Edmondson in Sir John Kendrew's laboratory. Using sperm whale myoglobin as a model protein is a tribute to its use in the first protein crystal structure solved in 1958. Our second protein is an unpublished crinarchaeal flap endonuclease. Crystals obtained from this protein grow with defects, and we hope to be able to reach near atomic resolution by screening and optimizing conditions in microgravity.
Thank you, Victoria. We will now take questions about the Real-Time Protein Crystal Growth 2 experiment. Again, please press *1 to be entered into the queue. Victoria, I have a question. What are the anticipated outcomes of this project?
Yes, so we're hoping to be able to see, at first the astronauts are going to screen the protein, in which we will converse with them on what the next step should be in terms of monitoring or optimizing the crystallization conditions that we're using, in which we'll use a second apparatus to set up these crystals in. We hope that when they return to Earth, we see improved size and improved quality and improved crystal packing, and to be able to use this in the future to screen difficult to crystallize proteins in microgravity.
That's great. Why is protein crystal growth important in microgravity?
Yeah. So the convection currents and thermal shearing and unit gravity are what cause the crystal growth to be hindered. This can be due to the feeding of the crystal, the protein in the drop feeding to the crystal being irregular, or just convection currents in the drop. And so microgravity, these are reduced, and therefore you don't have these effects. And so they should grow nice and large, and the packing should be nice.
Thank you so much. And again, for reporters, don't forget, if you want to get into the queue, please press *1. Okay, and next we will go to Carrie Lee, Multi-Purpose Crew Vehicle Radiation System Manager at NASA's Johnson Space Center, who will discuss the Artemis Hera on Space Station, or AHOS. Carrie, over to you.
All right, thank you. All right, so the AHOS is the Artemis Hera on the space station, and HERA stands for the Hybrid Electronic Radiation Assessor. And so the Artemis Hera on space station will demonstrate the operational readiness of the Artemis II Hera radiation detection system, and is going to provide the Artemis flight control teams with a data stream similar to that's expected on the Artemis II mission. The AHOS operations and data from ISS will provide direct experience with the Artemis Hera system, as well as insight for flight control teams to more effectively utilize the information provided by the instrument. HERA is a distributed ionizing radiation detection system that is what will be on the Orion vehicle. The AHOS consists of 3 of those detectors, and they are orthogonal to each other. And each detector contains a pixel detection chip with over 65,000 pixels. Each pixel acts as a single ionizing radiation detector. And together, we can observe charged particle tracks as they pass through the individual pixels. So HERA is going to be integrated into the caution and warning system on the Orion vehicle, and will alert the crew in the event of any large increase in ionizing radiation as a result of a solar particle event due to solar activity on the sun. Our primary concern is with solar energetic particles that can be accelerated by active regions on the surface of the sun. There is usually a flare associated with this solar activity, but we want to keep in mind that flares are not the problem. A flare poses no danger to the crew. It is these solar energetic particles that are accelerated from these flaring regions that are what cause the ionizing radiation that we want to detect with the HERA instrument, or the AHOS. The sun produces many, many flares that do not accelerate particles. So just having a flare does not mean that there will be particles to be detected. So overall, AHOS's purpose is to test the HERA response in the space radiation environment within the ISS, and also test commanding to the HERA instrument that will be on the Artemis missions. Thank you.
Thank you so much, Carrie. We will now take questions about the Artemis Hera on Space Station. Please press *1 to enter the queue.
All right, and our next question comes from Mark Curé from Aviation Week and Space Technology. Your line is now open.
Thank you very much. Will this instrumentation that's going to the space station be inside? I think that's what you were referring to, or outside the space station. And as far as Orion, where would it be placed? And I'm also wondering, does this work at all for cosmic radiation, or is this strictly solar?
Yes, it will be inside of the space station. The AHOS will, and the HERA that's in Orion will also be within the habitable volume, or in the pressurized volume. it is able to detect all charged particles. So whether they originate from the sun or from the galactic cosmic rays, it can detect all of those.
Thank you so much, Carrie. Okay, we will now take any additional questions for any of our speakers. So please, if you would like to ask a question, please press *1 on your phone. And when you get on the line, please identify to whom your question is directed.
Our next question comes from Bill Hallward from CBS News. Your line is now open.
Thank you. Yeah, I just wanted to follow up, Nicole Wagner, if I could, real quick. I appreciated the explanation before, but I'm still curious about how the signals that are generated by light, the electric signals, how does the biological systems in the eye interface with the artificial retina so that those signals are transmitted? I mean, is it synaptic? Does the charge in the artificial retina, does that ionic charge then, how does that trigger the cells that carry the signal to the brain? And I'm sorry for such elementary questions, but it's fascinating and I don't know anything about it. Thanks.
Sure. So, to take a step back, in these patients that have retinitis pigmentosa or macular degeneration, once they lose their photoreceptor cells, the remaining cells, or your bipolar and ganglion cells, usually remain intact. So we will put our artificial retina directly interfacing with those cells. Now, what we're going to do when the implant is stimulated with light is it's going to pump ions. So in this case, it's actually pumping protons. So we're changing the local pH around the bipolar and ganglion cells. That change in pH is being picked up by receptors on those cells, and it's that change right there that is going to send a signal to the optic nerve. So it's really, you can think of this as a receptor, mediated response, where that change in pH is picked up by acid-sensing ion channels, and then once those are stimulated, that's what sends the neuronal signal to the ganglion cells and then to the optic nerve.
And that's exactly what I was looking for. Thank you.
You're welcome.
Thank you. I have an additional question for you, Carrie, about the AHOS. What role is the space station playing in preparing NASA to go back to the moon?
From an AHOS perspective, this is preparing us to test the Hera system. So this is an identical piece of hardware that will be on the Orion vehicle. It will allow us to test the Hera response in the space radiation field, which is not easy to duplicate here on the Earth. And it will allow us to test the commands commanding Hera through the standard flight control system.
Thank you so much. And I wanted to give Heidi an opportunity to also add into that answer as to what role is the space station playing in preparing NASA to go back to the moon. Heidi, did you have any inputs on that?
Yeah, thanks for the question. I think ISS overall is a test bed for future exploration. So, a lot of our experiments, as we've talked about today, are geared toward improving our lives on Earth. But a lot of them are also geared toward helping us to expand our capability to explore future, to go onto the moon and to Mars and to further locations as well. And there's still a lot that we have to learn, that we need to figure out before we can effectively go to someplace beyond low Earth orbit. The nice thing about having the ISS in low Earth orbit is that, it's relatively easy to send up new supplies if something breaks, or, to resupply our food and water and consumables. But once we're talking about future exploration to someplace outside of our orbit, a lot of those things that we take for granted now in the ISS are not going to be possible to do in a location that's further out. And so, the more that we can learn now, the more effective and capable we'll be able to to explore further in the future.
Thank you so much, Heidi. And I've got one more question, actually. I'm curious for Dr. Vanapalli. On the Micro 16 investigation, how did you settle on the design that you used, the pillars, for measuring strength? Were there any other designs that you tried?
Sure, yeah, I can answer that. So I guess in terms of the design of the device itself, we basically spent almost a decade in terms of initial idea to actual execution of the optimized design. So the animals are crawling through these arrays of these pillars. So it's very important to make sure that the actual spacing between these pillars is optimized to ensure that they're able to actually push. Since in that sense, we basically had to do some initial iterations, and we also had to do some experiments to actually make sure that the animals are getting enough contacts with these with essentially all these pillars, and then we also looked into testing animals which are older and younger to make sure that because the animals grow in size, so as a result, it's important to accommodate the changes in the animal body shape and size. So all of those aspects were actually taken into account in terms of obtaining this optimized design.
Thank you so much. And I'm just very curious, and I've got one more question, and this one goes to Dr. Fernandez, if you're still on the line. With regard to the spaceborne computer, what type of experiments do you anticipate being able to work with, as we've heard you talk about some lightning experiments, among others?
Yeah, I am super excited that we don't have any experiments planned for ourselves in terms of computer science or anything of that nature, that we're opening it up to the community. And my goal is that we'll have tens or hundreds or maybe thousands of experiments that we can help other researchers advance their missions. So that's going to include radar processing, image processing, potentially DNA sequencing. We've put up state-of-the-art computer hardware that is available in all of these various disciplines. So we're pretty comfortable that we'll be able to address any of the processing needs that are brought to us. So thank you.
Well, thank you, Dr. Fernandez. That concludes our questions for today. I know I learned a lot. I hope you did as well. Thank you for joining us and your interest in this important science, research, and technology that is heading up to the International Space Station on the next Northrop Grumman cargo resupply mission. I encourage you to read more about our featured investigations in a story that's online now at www.nasa.gov/station. Stay tuned to nasa.gov/Northrop Grumman for more details about the upcoming launch and coverage on NASA TV. You can follow along with our research in these studies in orbit online at nasa.gov/ISS-science. And one more reminder, you can listen to a replay of this teleconference after about an hour by dialing 800-925-0147. One more time, that's 800-925-0147. The replay will be available after about an hour from this conclusion. Thank you again for joining us.
And that does conclude today's conference call. Thank you so much for joining. And of course, you may disconnect at this time.